Hi, my name is Maya Irwin, and today I'm going to be discussing a research article about biological control of mealybugs in China using green lacewings to find out which one does it better. This is a subject near and dear to my heart as somebody who works in a greenhouse and has to deal with mealybugs quite a fair bit. So now a quick introduction into the research article. This paper looks at sea salt, which is native to Mexico and was first cultivated by the Aztecs, but later would be cultivated all across the world. It's used for the production of rope, baskets, and even cat scratching posts, as it's highly valued for its strong and robust fibers. So what's the problem at hand? The gray pineapple mealybug has been devastating the sea salt industry in China. Not only does it cause direct damage to the plants with feeding, it's also a vector for pineapple mealybug wilt-associated virus that can cause further economic losses. On top of this, chemical control for this mealybug species has proven to be rather difficult. There are some biological control options, however, there are no natural enemies within China to manage this mealybug population. So this research article is seeking to find green lacewing species that are native either to China itself or nearby in order to manage this pest problem. So who are our saviors in this? Our first option is Plesiochrysia ramburi. P. ramburi has been spotted in China for the first time rather recently. It is native to Australia and neighboring islands, but it has also been spotted in Southeast Asia and Japan. And notably, it has been recorded to have preyed on other mealybug species before. Our second option is Apiterochrysia rosea frontata, which is quite the mouthful. This species is actually very recently described, which is exciting. It is a native in South China, but unfortunately there has been no recorded predation on mealybugs. And now on to methods. The first things that researchers wanted to look at was functional response. Larvae were collected from a sisal field and raised separately alongside the mealybug. Mealybugs were of the second and third instar as well as adult females, but not males. Lacewings were of first, second, and third instar, but not adults. Lacewings were placed inside of a petri dish container with a sisal leaf and a set number of mealybugs to record predation at different densities. With the information from the previous experiment in mind, a greenhouse was filled with sisal plants, and on each was placed 300 female mealybugs. Lacewings had been placed 3 days prior and provided Paralidae larvae as food, a Lepidoptera family. For an inoculative release, a 1:20 predator-prey ratio was used, and for an inundative release, a 1:10 ratio was used. The mealybug population was recorded over a 2-week period to measure predation. And now for results. P. ramburi showed a type 2 functional response with the second instar, third instar, and adult female mealybugs in all of its larval instars. Predation increased with age for both species. The first instar, Rosea frontata, however, showed no response when preying on third instar and adults, but it did show a type 2 functional response with second instar prey. Second and third instar of the lacewing larva displayed a type 2 functional response for all prey instars listed. And now for conclusion and discussion. The research concluded that the lacewing species tested exhibited a type 2 functional response typical of other green lacewing species. Both ramburi and Rosea frontata species showed predation on the mealybugs, with their third instars being effective most. Rosea frontata, however, did struggle more with handling prey and were thus often hindered by the defensive secretions of the mealybug. Researchers also found that a 1:20 ratio was economically optimal. Ramburi, however, has the edge, making it the clear winner in this competition. While these results are promising, there are still areas of further research. Authors indicate that due to the high prey density of their experiment, there should also be tests being done on lower prey densities. They also face spatial limitations, and due to this, their sample size was smaller than they would have preferred. Future research should ideally have a much larger sample size. Mealybugs are also sometimes protected by ants in the wild. for future research and for future application in fields, this should be factored in. For those wishing to read more, a citation is provided on this slide.